Hi everybody, it's Dr. Biology here, and I thought you'd like to see what a live lesson looks like. So many of us teachers at the moment are teaching from home, and obviously many students are learning from home. So I thought I would share an example. So this is an hour-long lesson, and I've just condensed it down into about 10 minutes. So it's useful to see what kind of things we do. So I hope you find it interesting. Right, okay, I will start to take the register and then we'll get started. Enough of that, let's get on with the lesson then. So what are we going to do? Well, we were talking about selection, directional, and well, different types of selection. So we're just going to recap that before we start the required practical. Now, obviously, it's very difficult to do a required practical whilst you're at home, so we're going to have to improvise. Hopefully, we will do this properly when we get back, but I do want to go through it. And it's very, very similar to the one you did at GCSE biology. So it should be coming through. Yep, it's there. I can see it. I'll just share that actually. So stabilizing and directional selection. So what I'm going to ask you to do is I would like you to get a sheet of paper and make it landscape, and I'd like you to put 2 circles on it like that. Okay, and then we're going to go through what we call the similarities and differences of the different types of selection. Okay, so I'll let you just get that together. Right, okay, so in the chat function, I would like you please to put down some similarities in terms of the types of selection there. There are things that are similar between them. Okay, so could you write your ideas please down in the chat function? We'll go through it anyway. Okay, so in terms of the similarity, so first of all, both are types of natural selection. The range does narrow, okay, and only some features get selected. Okay, so very much what you said. So both have something to do with phenotype. They do have something to do with phenotype. They do act against the extremes and they reduce variation. Okay, well, yeah, the range narrows. You only get some features selected. So yeah. Okay, so if you write that down, please. So that's what we call the similarities. And then we need to look at the differences. So we'll go through stabilizing selection. So the first is with stabilizing, both extremes get selected against. Okay, so both extremes, either side of that bell curve, if you remember, and it gets narrower, so the range narrows. In fact, I found I could do this the other day. So, ooh, I can draw a graph. Look. So what happens is that the range narrows. So shall I change the color? Pink color. So what happens is that gets eliminated and that gets eliminated. So you get a narrower range. Okay, so both extremes get selected against. Next one. Oh, I can't do it. Right, yes. It's not due to environmental change. Okay, so it's not due to environmental change. And thirdly, the mode does not change either. Okay. So the modal value won't change, but the extremes get selected against. Right, so directional selection, well, it's the opposite of stabilizing. So whereas in stabilizing both extremes get selected, only one extreme gets selected against. So for example, if you remember the giraffe, the short neck is getting selected against for the longer neck. Okay, so you get a movement of the curve to the right in terms of the neck length gets longer. Directional selection does tend to happen due to environmental change. So when there's environmental change, so you either adapt or die, that kind of thing, survival of the fittest. And the mode does change. Okay, so I'll give you a moment to write those down. It's quite a good way to remember the similarities and differences. It's called a double spray diagram, and you can do that with lots of things in your biology spec where you want to look at similarities and differences, and it's quite a good visual way of looking at that data.
What would be the title of this lesson? Sorry.
We haven't got to the title of this lesson yet. I'll tell you in a minute. This is just a kind of a recap. Okay?
Okay.
Right, okay. So you might remember as well, I'll leave that up for a minute. You might remember as well that we talked about antibiotic resistance. Okay, so we talked about antibiotic resistance. What form of selection is antibiotic resistance, please? So in the chat function, what form of selection is it? Yep, it is directional. Be more confident, Rosie, you are correct. Yes, directional selection. So alleles for characteristics of an extreme type are more likely to survive and reproduce. And we talked about antibiotic resistance. Now, obviously, why do you think it's really important for biologists to study antibiotic resistance? Why on earth would we want to study that type of work? So why would we want to study this? Yep. So we might want to make new antibiotics. Very good. We might want to do some experiments to see how fast a certain bacteria might be becoming resistant, okay, to stop bacteria in the future becoming resistant as well. And what we're going to need to do is to investigate this, we need to investigate the effects of antibiotics on bacterial growth. Okay, and that is what we're going to look at. And we're going to look at today about how you investigate that, but also how you do what we call aseptic techniques. So investigating the effects of antibiotics on bacterial growth. Okay. So what do you think aseptic means? So in the chat function again, what does aseptic mean, please? What do we mean by aseptic? Yeah, no bacteria. Good. Any others? Basically, it's free from contamination caused by harmful bacteria viruses or other microbes. So it's what we call surgically sterile or sterilized. So that's what we mean by aseptic. So we can achieve aseptic environment in the laboratory. We're going to learn about this. Well, you can work close to a Bunsen burner using flaming loops and bottles, keeping Petri dishes open for a minimum duration. Okay, and then antimicrobials that you would be studying are things like antibiotics, bacteriophages, essential oils, disinfectants, stuff like that, bleach even. Okay, so there's some really good definitions there. So I'd like you to write those definitions down, please. So yeah, so we'll do another Jamboard, and we're going to look at the risks, the hazards, and how you take precautions to reduce those hazards. Also, this person is not me, you'll be glad to know. It's an actual microbiologist who knows what they're doing. And in fact, I couldn't do it here, could I? Oh, here's my Bunsen burner. Oh, I'm going to burn the house down. So yeah, you done now, Toby?
No, but I can take a screenshot.
Okay, take a screenshot. That'll be great. Just give you a moment. So yeah, this is about a 5-minute video. So I'd like you to look at it carefully. And also, I particularly really want you to look at the techniques that this person is using. Okay. Sorry? You done?
Yeah.
Great. So, okay, so I'll share this video with you. I'll have to turn off my microphone. So, see you in a bit.
Your plates will look like this. You can see the areas where bacterial colonies have grown. Some of the antibiotic discs will not have any bacteria growing around them. These zones of inhibition indicate that the antibiotic has killed or inhibited the growth of this bacterium.
Okay, good. Okay, so hopefully you can see that. It was a bit glitchy for me, but yeah. Anyway, so she was going through all of the kind of aseptic techniques you would do to produce a culture of E. coli. So E. coli is a species of bacteria. So we need to note down a few things. So that's what we're going to do now. Precautions. Save. Can you access it now? Yay, you can. Good. Great. Okay, I'll leave you for a minute to put your answers. Don't make your post-it notes too big. Great, loads of ideas. Brilliant. Can the getting ill be a bit smaller? I know you tried to do it. Hey, did it. Good. So yeah, very good. So risks is the Bunsen burner, bacteria escaping, okay, causing a pandemic. It's possibly not, but there you go. Oh, I think it's because I've still got my Jamboard open. Wait a minute. So I'm going to close Jamboard. There you go. So present window. There you go. There it is. Right. So in terms of safety, Okay, so we talked about Bunsen burners. So they're yellow flame when not in use. Long hair has to be tied up. Okay, ethanol is used to clean inoculating loops, highly flammable. E. coli, the bacteria, you need to make sure they're properly sterilized. The big one is if agar plates are completely sealed, hazardous anaerobic bacteria may grow. Therefore, agar plates are closed with, well, I would say 3 pieces of sticky tape. Okay, so the top 4 bullet points, I would like you to copy down, please. So just the top 4, not the other 2. Assuming everyone's done that. Okay, right, so just go on to the next couple of slides. So we need to talk about general principles. Okay, now I am going to tell you ways of making this a bit shorter. So the first thing before you do any aseptic techniques, you have to disinfect the work area. And as I said, you use ethanol. You can use ethanol. And also you need to, after you've finished, you need to protect others as well from possible contamination. So we're going to go through 5 general principles. So if you could write that down, please. Second one, and we can shorten this. Okay, we're going to shorten this. So the inoculating loop is the thing that you spread the bacteria with. Now that the microbiologist in the video didn't show us that. Okay, so it's how you transfer bacteria. So all I would like you to write is flame the inoculating loop, never put it on the bench top, never lay it on the bench top. When in doubt, flame the loop. So the open glass container. So you might remember she had a glass container and she flamed the top of the bottle. Okay, so the opening of the glass container before removing the bacteria. So all I'd like you to do is write the first sentence. So it says flame the opening of glass containers before removing bacteria. There you go. So that's all I would like you to write. So just flaming the top of the bottle. And then you would have noticed the microbiologist did the following step and we can shorten this again. So don't lay the cap of the back of the container on the bench top. Okay, so basically you should just keep hold of the cap top. The reason being is you can, if you put it on the bench top, it will be contaminated. Okay. So if you just want to say hold the cap of the container okay, and finally, number 5. So work quickly and efficiently to minimize the time the culture is exposed to the environment. So for example, keep only opening the lid slightly. Okay, give you time just to write that down. Right, okay, so you've done that. Yeah, has everyone done that? I will put it on Google Classroom as well. Yeah, thank you. Right, okay, so just going to show you a couple of questions here. Okay, and I'd like you to have a think about these questions, and then I'd like you to put your answers in the chat. So I'll give you about 4 minutes or so to do this. The reason I've given you 4 minutes is I need to check why my dog is going mental downstairs. So I'll be there in a minute. Okay, right. So some good answers there. So we write our name on the underside. Yeah, it is because it's placed upside down. So when it grows, one of the key things though is if you put it on the lid, the problem with putting it on the lid is if you're using multiple samples, what can happen is that you could place lots of lids down on the lab bench, and they could be mixed. So you wouldn't know which lid went with which base. So if you write everything on the base, it means that if you misplace the lid, you won't lose the whole sample. But yeah, it's upside down when it's growing, definitely. Why do we flame the McCartney bottle? Because Ringo tells us to. No, that's a really bad joke. The bottle, the reason we do is so that it becomes sterilized and you don't contaminate the culture with a different type of bacteria. Very good. Prevent contamination. And what is the difference between disinfectant and sterilized? Well, a disinfectant is actually a chemical that is used to sterilize things, and sterilized means that it is microbe free. So there's no microbes at all. So it's completely clean, sterile, and aseptic. Okay, right. So next lesson, I will be showing you some results. We'll be looking at inhibition zones. Now, I think you've heard of inhibition zones at GCSE. You should have done if you did GCSE biology. We'll talk about some results, and then we're going to look at some questions related to selection and antibiotic resistance. Okay, so I'm quite happy to finish there. Yeah, we did. You remember, Elliot? Yeah, we actually did that, I think, in year 9, possibly even. I'm not too sure. Anyway, okay, so thank you very much, everybody. If you have finished, I will put this on the Google Classroom now-ish, once I've stopped the dog from going mental. Any questions? No? Right, you can go then. Thank you very much.